Hello everyone, my name is Selena and today I'm going to give a short presentation on my project by engineering bacterial outer membrane vesicles as delivery systems for RNA therapeutics targeted to lung epithelial cytosolic. I'm going to give a brief introduction of the project. I'm going to present the hypothesis and objectives. I'm going to present the results generated within the 3 years of my PhD, and I'm going to state the conclusions and the future studies we attempt to perform. As it is widely known, there are many diseases which are associated with lung tissue. It has been shown that topical administration is leading to inefficient cell uptake. Scientists have shown that inhaled administration of oligonucleotides causes non-specific targeting, as the labeled oligonucleotides do not enter lung epithelial cytosols, and they enter systemic circulation instead. Moreover, another issue is the inflammation and how the macrophages respond to delivery systems. The solution is to generate targeted delivery systems derived either from natural particles such as viruses, including adeno-associated virus and lentivirus, or by exosomes, which are viral-like particles. However, they cannot be replicated. They are difficult to manufacture at scale, and they are associated with cancer signaling. They might drive genetic drift risk due to their genetic background. On the other hand, scientists have generated synthetic delivery systems by using receptor transport mechanisms or particles with bioadhesion properties, such as cytosine particles, or combination of them. However, they are very complicated to be manufactured and production costs are very high. Our approach is the outer membrane vesicles, or OMVs, for all the interval purposes. They are the bacterial version of exosomes. As a reminder, bacteria like eukaryotes have two double-layer membranes, one of which is called the cytoplasm and the other one is called the periplasm. So bacterial outer membrane vesicles are formed from the periplasmic membrane of bacteria. They have sizes from 20 to 250 nanometers. They are produced from gram-positive and gram-negative bacteria, whether they are pathogenic or not, and they contain DNA, RNA, proteins, and metabolites. In terms of biological function, they have been associated with toxin removal, immune evasion, immune bioadhesion, and nutrient shuttling. But more importantly, and relevantly to us, inter-kingdom crosstalk. In other words, they shuttle molecular signals from bacteria to eukaryotic cells. What is most advantageous about outer membrane vesicles as compared to exosomes is that they have a highly defined isolation methodology, which involves a few steps of filtration, either through small filters or tangential flow filtration. Maybe ultracentrifugation can apply as well. We know that we can remove toll-like receptor agonists such as LPS by using LPS-deficient packaging systems such as clear coli, creating stealth OMVs, and we know we can produce an established manufacturer protocol that is clinically and commercially viable. The MenB vaccine, which is the most used vaccine globally, is based on outer membrane vesicles from Neisseria meningitis. So our question was, if these stealth OMVs can package bioactive compounds, and if we can use them with transmembrane proteins to deliver bioactive compounds to eukaryotic cells. So we have decided to generate lung-targeting clear coli to produce fluorescently labeled outer membrane vesicles and to try and demonstrate proof of principle in vitro. So approaching these, the aim was to create a lung epithelium targeting clear coli. So this is the E. coli that produces stealth OMVs, which are fluorescently labeled, and to use these vesicles to demonstrate the principle in vitro. To target the lung using outer membrane vesicles explicitly, we used a proprietary transmembrane protein. Additionally, to label the vesicles with fluorescence protein, we decided to use a protein fusion strategy. So we fused the transmembrane protein with a dual RFP reporter protein, Tindy tomato, either at the N terminus of the protein or at the C terminus. We used the PET28A vector for this, whose multiple cloning sites include EXO1, ECOR1, and ND1, and the two cloning strategy to introduce first the transmembrane protein and then the RFP on the correct orientation. In the gels on your right, you can see that we cleaved up the clones with the correct inserts, approximately 1,500 bases for tinted tomato and 800 bases for the transmembrane protein. Once the protein was recombinantly expressed in the clear coli, we first attempted to detect whether transmembrane protein is produced. And what we can see on the first gel is that the transmembrane protein is produced. And what you can see on the first gel is that the transmembrane protein tagged with has a molecular weight of approximately 75 kilodalton is found the correct molecular weight on the tagged construct, and not in our negative control coli, but is exclusively in insoluble fraction. On the second gel, you can see that the fusion protein is detected by anti-TMP in membrane fraction as well. So we used the anti-TMP antibody to detect the fusion protein in the outer membrane vesicle fraction, and we observed that the transmembrane protein was found only on the N-terminally tagged outer membrane vesicles and not on the C-terminally tagged vesicles. Interestingly, this protein was back at its original molecular weight, i.e. 30 kilodaltons, suggesting that the cargo has fallen off the membrane protein. So we decided to proceed with the N-terminally tagged outer membrane vesicles. Interestingly, we found that when expressing a soluble protein like tinted tomato in E. coli, only 1% of that protein ends up in the outer membrane vesicle fraction. However, if this protein is placed at the N-terminus of our transmembrane protein of interest, the amount of protein that ends up in the vesicles increases to 7% of the recombinant protein. In other words, we have a 7-fold increase in packaging efficiency for our recombinant soluble protein. We then proceed to carry out transmission electron microscopy, and we observe no drastic differences in the sizes and morphology of the vesicles, whether or not we had our control OMVs. OMVs labeled with RFP only, OMVs with recombinantly expressed TMP, or OMVs with N-terminally tagged transmembrane protein. As you can see, from the nanoparticle tracking analysis, the size distribution is comparable across the 4 formulations. We have also decided to continue to check the proteomic profiles between the OMVs, and we observed that they were highly consistent. We performed principal coordinate analysis and unsupervised clustering. We observed that there were 1,100 different proteins within these samples, from which approximately 400 differ by OMV class. So it means that the structure of OMVs changes, but in a constant way. Hereinafter, we attempted to study the effects of outer membrane vesicles in A549. For this critical experiment, we used 10,000 A549 cells per well, which were allowed to adhere overnight, and we seeded the cells with 10,000 vesicles per cell. We allowed binding to take place for 1.5 hours at 4 degrees, and we observed that internalization is maximum within 30 minutes at 37 degrees. These data are from low magnification, 40 times fluorescence microscopy, where we can apply unlabeled outer membrane vesicles and TMP outer membrane vesicles. And on the right picture, you can see the OMVs containing RFP only. You can strangely see that we have district delivery of tinted tomato in A549 cells with the N-terminally tagged outer membrane vesicles. But with the RFP OMVs, we have only few of the cells that, in which the fluorescence is detectable. These data are representative of 3 independently carried biological experiments carried out independently in 2 separate labs, one in the UK and one in Germany. So to summarize, we have shown that clear coli would generate stealth OMVs, which is not recognized by toll-like receptor 4, can tolerate an epithelial cell targeting transmembrane protein being expressed in its OMVs. And we have shown that by N-terminally tagging this particular transmembrane protein increases the loading of OMVs by 7 times. We've also showed that the transmembrane protein dissociates from this particular N-terminal fusion in OMVs, and that their cargo is in the lumen. We have evidence from early in vitro efforts that this particular transmembrane protein improves the targeting of OMVs to type 2 alveolar epithelial in vitro. Next steps are to validate in air-liquid interface primary cell culture, confirm cargo subcellular trafficking mechanism, test in vivo delivery, and to generate therapeutically relevant cargo system. Finally, I would like to thank my supervisor, Prof. Sergios Moschos, for his support through this project, and my second supervisor, Professor Gary Black, for his scientific advices. Also, I would like to thank my colleagues at Northumbria University and our collaborators at the University of Hull. Thank you very much.